Bismillahirrahmanirrahim. Assalamu alaikum dear students. I am Razia Bashir from University of Education, Lower Mall Campus, Lahore. I am co-teaching the subject of General Biochemistry with Dr. Muhammad Tariq from Division of Science and Technology, Township, Lahore. The topic which we are going to discuss today is the fate of pyruvate. In our previous lecture, we have discussed that a glucose molecule is broken down into two pyruvate molecules through the process of glycolysis. It is a series of enzyme catalyzed reactions. At the end of these, we produce two molecules of pyruvate. Today we will discuss the fates of pyruvate, which is actually the fermentation. Glycolysis releases only a small fraction of the total available energy of the glucose molecule. At the end of glycolysis, we obtain two molecules of pyruvate, which still contain most of the chemical potential energy of glucose. Energy that can be extracted by fermentation or Krebs cycle. In most of the anaerobic organisms, after glycolysis, the pyruvate enters into fermentation process either through the alcoholic fermentation or lactic acid fermentation. In aerobic organisms like humans, in some situations due to lack of oxygen, they also process pyruvate through the lactic acid fermentation. While in aerobic organisms, most of the pyruvate enters into Krebs cycle, which is actually the oxidative phase of pyruvate breakdown. In this figure, you can see three possible catabolic fates of the pyruvate which was formed in glycolysis. This pyruvate can also serve as a precursor in many anabolic reactions which are not shown here. However, we will discuss these anabolic pathways in gluconeogenesis. Over here, we will see the catabolic fates of pyruvate. Pyruvate is produced from a glucose molecule through a series of reactions. This pyruvate can be processed under two conditions, either the hypoxic conditions or aerobic conditions. In the hypoxic situation or anaerobic conditions, a pyruvate is broken down into two molecules of ethanol along with the two molecules of carbon dioxide. This is called as ethanolic fermentation, which mostly occurs in yeast. The second process of pyruvate catabolism, which is anaerobic, is the lactic acid fermentation, in which a pyruvate is converted into lactate in vigorously contracting muscles, in erythrocytes, in some other cells, and most of the microorganisms. While in aerobic conditions, pyruvate is converted into acetyl coenzyme A by releasing two molecules of carbon dioxide. This acetyl coenzyme A enters into mitochondria, where it is processed through the Krebs cycle or citric acid cycle, and this is common in animals, plants, and many microbial cells which have the aerobic conditions. Fermentation is a general term which is used for those activities, those processes which extract energy in the form of ATP but do not consume oxygen or change the concentrations of NAD or NADH in cellular systems. Fermentations are carried out by a wide range of organisms, mostly by microorganisms and yeast. Most of these organisms, they live under anaerobic conditions, and they yield a variety of end products through the fermentation reaction. Some of these products are very valuable for commercial uses, such as cheese, margarine, etc. These are produced by the fermentation reactions at industrial scale. In living organisms, there are two basic forms of fermentation. First one is the lactic acid fermentation. It occurs in most of the organisms under anaerobic conditions. For example, when animal tissues cannot be supplied with sufficient oxygen to support aerobic oxidation of the pyruvate and NADH produced in the glycolysis. In that situation, NAD is regenerated from NADH by the reduction of pyruvate to lactate. For example, when humans do extensive exercise in their muscles, there is lack of oxygen. In such situation, usually the pyruvate is processed through the lactic acid fermentation. Some tissues and cell types, such as erythrocytes, which don't have mitochondria and thus they cannot oxidize pyruvate to carbon dioxide. These tissues and cells they produce lactate from glucose even under aerobic conditions. The enzyme which is used for this process is lactate dehydrogenase. This is the simple reaction which takes place in lactic acid fermentation. In this reaction, pyruvate is reversibly converted into lactase in the presence of lactate dehydrogenase enzyme. And in this reaction, NADase is converted into NAD. The lactate, which is formed in the process of lactic acid fermentation in active skeletal muscles or erythrocytes, can be recycled. It is carried in the blood to the liver, where it is converted to glucose during the recovery from strenuous muscular activity. When lactate is produced in large quantities, as during vigorous muscle contraction, the acidification that results from ionization of lactic acid in muscles and blood limits the period of vigorous activity, resulting in muscle fatigue. Second type of fermentation in living organisms is ethanolic fermentation. Yeast and some other microorganisms they ferment pyruvate to ethanol and carbon dioxide rather than lactate. Glucose is converted to pyruvate by glycolysis and the pyruvate is converted to ethanol and carbon dioxide in a two-step process. In the first step, pyruvate is decarboxylated in an irreversible reaction which is catalyzed by pyruvate decarboxylase. It means the same enzyme cannot go back or cannot produce pyruvate from ethanol. In the second step, acetaldehyde is reduced to ethanol through the action of alcohol dehydrogenase. This is the overall reaction of ethanolic fermentation, starting with the pyruvate, which is converted to acetaldehyde by the reaction of pyruvate decarboxylase enzyme, and in this process, a carbon dioxide molecule is also released. In the second step, acetaldehyde is converted into ethanol in a reversible reaction in the presence of alcohol dehydrogenase, and in this step NADH is also oxidized to NAD. Alcohol dehydrogenase enzyme, which is used in ethanolic fermentation, is present in many organisms that metabolize ethanol, including humans. In human liver, it catalyzes the oxidation of ethanol, either ingested or produced by intestinal microorganisms, with the concomitant reduction of NAD to NADH. This is the end of our today's discussion. Thank you for listening and learning it carefully. If you have any question, you can ask your question through the WhatsApp group or Google Classroom.